in 2020, colon cancer was the third most diagnosed cancer in Canada, and it's often referred to as the silent killer because it isn't till the late stages when it's fatal that it's finally diagnosed. However, with early diagnosis, it's actually one of the most curable cancers. So current research shows that metabolites can present themselves in high levels in our urine and be identified as biomarkers for colon cancer. And we've focused our studies on 3 different metabolites. They are hippuric acid, diacetylspermine, and creatinine. So I'd like to acknowledge some members of the Wishart Lab Group who have been working on this project, specifically the PI, Dr. David Wishart, and the electrical engineer on this project and CEO of Trica Technologies, Dr. Scott Mackay, and the chemist on this project, Dr. Yagana Kaniani. So we operate out of the University of Alberta and are funded by Trica Technologies and the Nanomedicines Innovation Network. So mainstream approaches to diagnosing colon cancer include colonoscopies and blood tests, both of which are extremely invasive. So we want to develop a test that is more accessible and less invasive in the form of a biosensor. So the biosensor will test for the metabolites mentioned previously in urine samples, and they will use nanoparticles that are decorated with these key metabolites and antibodies bound to an electrode. It will be in a competitive assay where these nanoparticles will compete with free metabolites in a patient's urine sample to bind the antibodies. This will cause an electrical change, which is an impedance reading, and we can use this to determine the concentration of these metabolites in a urine sample. So we focus on 2 types of nanoparticles, gold nanoparticles and liposomes. On the left is an image of our gold nanoparticles, which are synthesized in the lab, and on the right is an image of liposomes, which are provided by the Nanocore in BC. So as you can see, they have very different characteristics from size and color and conductivity, and we can utilize the difference in these characteristics to determine which nanoparticle will function best as part of the biosensor electrical system. So to bind these metabolites to the surface of our nanoparticles, we use linker molecules composed of polyethylene glycol, which is shortened to PEG. So with gold nanoparticles, we initially bind this linker to its surface, which not only helps bind the metabolite, but also provides stability to the nanoparticles by restricting them from binding together and aggregating. So then, in order to bind the metabolite, it's necessary that we form an amide bond. So when the linker has a carboxylic acid present, it's also necessary that metabolite has a primary amine and vice versa for binding. So as for liposomes, we use a phospholipid PEG linker, where the phospholipid PEG linker is compatible with the membrane of the liposome. So first we bind the metabolite in the linker, and they form micelles in solution. Then these micelles are incubated with liposomes at 60 degrees Celsius to raise the phase transition point so that we're able to anchor these linkers and metabolites into the surface of the liposome. So we characterize gold nanoparticles using ultraviolet visible spectroscopy. This is because gold nanoparticles exhibit a phenomenon called surface plasmon resonance. This is where they absorb light in the green-blue portion of the visible spectrum and reflect red light, which gives them that dark red wine color. So as you can see here, gold nanoparticles prior to any conjugation reactions exhibit an absorbance maxima of 518 nanometers, and this steadily increases after binding the linker and the metabolite. So this has been correlated with an increase in particle size, which is an indicator to us that the linker and the metabolite have been bound. Another technique to analyze our nanoparticle samples is dynamic light scattering. Here we're able to gather the relative hydrodynamic size as well as stability as indicated by the zeta potential. So Our liposomes, just bare liposomes, have a hydrodynamic size of 93 nanometers with this technique. And you can see this increases after post-insertion of the linker and the metabolite. As well, with the more negative the zeta potential value, it's an indicator of how stable our sample is in solution and less prone to aggregation it is. So to verify that the metabolite has been bound to our linker, specifically metabolite creatinine, we performed a colorimetric creatinine assay. And the function of this assay is that free creatinine forms a complex with picric acid and forms a chromogen that we can then measure the color intensity at 490 nanometers. So initially we tested our linker molecule that had creatinine bound to it, and we saw no color change. However, once we hydrolyzed our sample and broke apart that amide bond, we were able to see this wavelength increase greatly, or sorry, this intensity increase greatly. So that indicated to us that the metabolite was present and also bound. And we also, another way to determine the presence of acetylspermine, we used an ELISA. And the mechanism of this ELISA is that the free metabolite was bound to the bottom of a well. And when we tested our blanks, the primary antibody, instead of binding our blanks, because there were no metabolite present, the primary antibody bound the free metabolites at the bottom of the well. Then during the washing step, the blanks were discarded and the secondary antibody bound the primary antibody, and we got a color change. Now, when we tested our samples, the primary antibody bound our samples because the metabolite was present. So this meant that the primary antibody and our samples were discarded during the washing step, and the secondary antibody was unable to bind anything. So this is shown really well with our liposome results, where our bare liposomes are very dark in solution and have a very high intensity of the color change, while our liposomes with the metabolite bound to the surface are nearly colorless in solution. And we can see this difference in intensity as well with our gold nanoparticle samples. So all of these characterization and conjugation steps are necessary as we work to the next step, which is the development of the biosensor. We will use an interdigitated electrode that has a silicon dioxide substrate, and this surface will be modified prior to binding the antibodies. Then we will bind our nanoparticle metabolite samples to the electrode, and this will cause an impedance reading based on this nanobody and antibody binding. Then when we add a urine sample to the electrode, any free metabolites present will unbind our nanoparticles. This is because the free metabolites have a higher affinity for the antibodies than nanoparticles do. This will cause a change in impedance, which we can then measure and determine the concentration of these free metabolites in a urine sample. And example results are shown below, where gold nanoparticles were used to detect leucine residues. And here with this graph, we can focus on the impedance magnitude curves, which are the blue and green curves, where green represents no gold nanoparticles bound and blue represents gold nanoparticles bound to the electrode. And as you follow along these curves, they begin to form a gap. And where this gap is largest is where we can determine the best frequency to measure this impedance change. So thank you for coming to my presentation. If you have any questions or want to reach out about the project, feel free to email me here. And yeah, I'll take any questions. Thank you.
Payton, was this where you expected, or when do you expect this project to be complete?
So right now we're working towards getting impedance values from the gold nanoparticles in liposomes. So just getting a proof of concept right now. However, it probably be another year in order to test with urine samples and as well as just compare and contrast between which nanobody will work the best in the system. So yeah.
Excellent. I do have a couple more questions. Payton, do you think gold nanoparticles or liposomes will work better for this biosensor?
Sure. So traditionally, gold nanoparticles have been used because of their metal surface, which is very conductive. However, liposomes are kind of a new nanoparticle to look at for this technique. Actually, I can share my slides. I have a I can share this.
Absolutely.
1 second. So what we're considering right now is that gold nanoparticles, while they are very conductive, they also are a little bit more difficult to work with because they aggregate quite easily. And as you can see here, the PDI, which is a measure of homogeneity of our sample, is quite high. So not as consistent when it comes to developing a medical test. However, liposomes have a quite lower PDI and are a little bit easier to prepare because there aren't as many considerations we need to make in terms of buffers and what will cause them to aggregate. And as well, next steps also include here are some liposomes that we've also received from the Nanocore, and ones we are considering and will work with soon are these liposomes that have gold nanoparticles encapsulated. So we're excited to see if maybe a hybrid will actually work best in the biosensor.
Great. Thank you.
Yeah.
Marshall, are there any other questions at this time?
Yes, in fact, we have a written question from Yu Tian, who asks, why was liposome chosen?
Liposomes we've known them for encapsulating drugs and things like that because of their biocompatibility. However, liposomes here just present for us a new avenue to work on just because they're a lot larger and more flexible. And especially with the opaque liposomes we've been working with, it makes verification techniques more available to us to determine the binding of these metabolites to the surface. So again, maybe opaque liposomes might not be used in the final steps of the biosensor because they're clear, but they do allow us to visualize really well if the metabolite has been bound to its surface.
Great. Thank you, Payton. And thank you for the question. Have similar projects like this been done to develop diagnostics for other diseases that you know of?
Yes, actually a published article was about folic acid detection with gold nanoparticles. It was published recently or a few years ago. And what they did slightly differently is that they used a folic acid binding protein instead of an antibody. However, they were able to detect folic acid based on the presence of this conjugated metabolite on the surface. And they also were able to determine that based on the amount of nanoparticles bound to the electrode, they could change that concentration to change their limit of detection and limit of quantification actually of that assay, which is really interesting.
That's very cool.